Hello, and this is the third video I'm making. So this lesson actually goes back. Usually I teach this lesson first, but this lesson is one of the easier ones to teach by video. So I decided to send this one for video over Thanksgiving break, as opposed to doing the replication one over Thanksgiving break. So this lesson talks about the experiments that were used to study that DNA was the genetic material. So first of all, you probably learned in 9th grade bio that DNA is the substance of inheritance. It makes your traits. Okay, so why you have blue eyes and blonde hair, whatever color blue eyes you have and hair you have, it's because of your DNA. Okay, your DNA contains, okay, all the instructions to make all the protein in your body. So you make about 30,000 proteins, okay, and the DNA contains the instructions to make those proteins, which cause many different types of traits and allow the body to do everything it needs to do. Like DNA polymerase is coded for by a gene or by DNA. ATP synthase, okay, is coded for by a gene in the DNA. But the purpose of this lesson is to go through the experiments that allowed scientists to find that it was DNA that was the genetic material. So, again, DNA is the genetic material, but early scientists actually thought protein, not DNA, was the genetic material. And the reason why they thought that is that they knew at the time that there were 20 different amino acids that make up protein, but that there were only 4 different nucleotides, 5 if you're including the U and RNA. Okay, so that existed. So again, there were 20 amino acids and 4 nucleotides, so A, T, G, and C. And remember, with the amino acids, there were 20 of them, but we have acidic, basic, polar, and nonpolar. And because there were many more amino acids, they thought that they could code for more ably all the traits. Okay? But it turns out that they were wrong. Okay? And that it was DNA that is the genetic material. And the experiments that we're going to look at study bacteria and the viruses that infect them. One thing that scientists knew at the time is they knew that viruses, when they infected bacteria, injected their genetic material into them. But they did not know what that genetic material was. So the first experiment we're going to look at, which is called the Griffith Experiment, done by Frederick Griffith, he did an experiment with 2 different types of bacteria. One strain of bacteria, which was called the S strain, was pathogenic, meaning that when it infected mice, it caused death. And in terms of what the bacteria looked like, it had a protein capsule surrounding it. Protein capsule is almost like a cell wall to some extent. The protein capsule allowed it to be pathogenic. Then, he also had R strain bacteria, which is a non-pathogenic bacteria, and it was non-pathogenic because it did not kill the mice when the mice got infected. And this R strain did not have a protein capsule, which didn't allow it to kill the mice. Okay, so now we're going to look at the experiment. Lots of little mice. Okay, so A and B were control tests. Okay, because they knew what should happen, and they just wanted to make sure it occurred. Okay, so he first infected S strain, which is the pathogenic bacteria, into the bacteria. So he injected the S strain bacteria into the mouse, and poor little mouse, he died. Kaiser would get him. Okay, so that was a control test, and it proved that the experiment was working because they knew that S should kill the mice, and the mice died. Okay, then as another control test, he injected R strain, which is non-pathogenic. Notice it doesn't have that blue capsule around it. Okay, and when he injected the mouse with that strain, it lived. Happy mousey. Okay. Then he injected heat-killed. That's why it's red. Heat-killed pathogenic bacteria. And if you remember, heat-killed, the thing is heat-killed, that means, it's like stuff is denatured, so it won't work. It also denatures the capsule, which is protein-based. And when he injected the heat-killed pathogenic bacteria into the mouse, it lived. And he also expected that, so that was a control. Okay? All these results were the expected results. Then, in the 4th experiment, which was the experimental group, he injected heat-killed S, the pathogenic one, with R together. And the mouse died. But why would that be the case? Here, the mouse lived. When you injected the heat killed alone, it lived. And when you injected the R alone, the mouse lived. But when you inject them both together, it dies? Huh? What Griffith concluded from this is that something from the heat-killed S moved into the R, giving it the ability to kill. So something, this is dead, so it can't kill anymore. The capsule is destroyed. But something inside the heat-killed S moved into R. Okay? And he called this phenomenon transformation. When the phenotype is changed. The phenotype is what the organism looks like, what the organism can do. And what he concluded, again, is something in the heat-killed bacteria, heat-killed S, transmitted into R the disease-causing properties. But he did not know what this transformation principle was. So, just to summarize that whole long spiel, the conclusion from the Griffith experiment is that there was some sort of factor in the S bacteria that was able to transform the R bacteria into being able to kill. Now, if we go back to what is the evidence that supports that conclusion? Well, the evidence is here. And if we look, the evidence that is important is that when R and S are put together, but S is heat-killed, the mouse dies. So what he concluded from that is that something from S that can kill when it's living must have gone into R to make R be able to kill. Okay, now, after this transformation factor was discovered, people wanted to really find out what it was. People started doing all these massive experiments. What is this factor that Griffiths just found? And the first experiment that discovered this factor was the Avery experiment. And in the Avery experiment used enzymes. That's a nice spelling of enzymes. Look at that, lovely. So that S should not be there. And used three enzymes in the experiment. And one enzyme he used was DNase. And DNase breaks down DNA. He used RNase, and RNase breaks down RNA. And he used protease, which breaks down protein. Okay, now, in his experiment, he used the same R and S bacteria that Griffith used. And he treated, so this is the S bacteria here, and he treated the S bacteria with RNA, and it was heat-killed, just like in the Griffith experiment, okay? And he treated it with RNase, which destroys RNA, and he put it with the R bacteria, just like in the Griffith experiment. And what he saw is that in this experiment, the R bacteria were transformed. You can see they have those protein coats, and they were able to kill mice. Then, he did the same thing, but he put the protease enzyme with the S-killed bacteria. And then he mixed it with R bacteria. And again, he saw that the R bacteria were transformed, or had that capsule that allowed them to kill. And then finally, he put the S-killed bacteria, or the S-heat-killed bacteria, and he put it with DNase. Okay? And then he mixed it with R bacteria. And he saw that in this case, there's no transformation. Okay? No transformation occurred. Okay? What he concluded from this is that since the bacteria are not transformed when the DNA is transformed, it must be DNA that is the genetic material. So the conclusion from this experiment was that DNA is the genetic material. The evidence is that only when the S bacteria, heat-killed S bacteria, were treated with DNase, only in that circumstance were the R bacteria not transformed. In the other two samples where we used RNase and protease, the R bacteria would be able to be transformed. So from that, they were able to conclude that it was DNA that was the transformation principle. Okay, we have one more experiment that we need to talk about, and that is the Hershey and Chase experiment. This is by far the most confusing one. So Hershey and Chase, Alfred Hershey and Martha Chase, set out to confirm the Avery experiment. And the question they asked is DNA the genetic material? Okay. The information known, okay, by Hershey and Chase was that viruses, which are also called phages, they infect bacteria by injecting their genetic information into the bacteria. So that was one thing that was known. Whatever the genetic information is, they inject it into bacteria. The other thing that they knew is that protein has the atoms C, H, O, N, and S, and DNA has the proteins C, H, O, N, and P. So if you take a look at these, S is unique to protein, and P is unique to DNA. After I talk about this, if you're still confused, this is a great animation of the experiment to watch here. Okay, so I do not want to run out of time in the middle of explaining this, or in the middle of explaining the Hershey-Chase experiment, so I'm going to upload this video and continue with the Hershey and Chase experiment in the second video. The URL for the second video, instead of tinyurl.com/turkeyvideo1, which was the link to get to this class, it's going to be tinyurl turkeyvideo2. I will see you there.